fertilization. So what is fertilization? Fertilization is basically a in humans refers to the fusion of male and female gametes. We know that the male gametes are produced in the testes of a male reproductive tract, and this female gametes are produced in the female reproductive system. Okay, that is ovaries. So what happens that the fertilization is a natural life process, and it takes place in the fallopian tubes. If we look at the this diagram, it is clearly shown here that the what is where fallopian tube is located inside the female reproductive tract. So this is the this is the fallopian tubes. It is coordinated with the ovaries. As we know that there is a pair of ovary at both sides of the female genital tract. Okay, so continue from the previous slide, that the as we know that the semen is comprised of thousands of sperms are inseminated into the female genital tract, and this female genital tract uh, continued with the thousand of sperms, and sperms move towards the uterus and reach the opening of the fallopian tubes. Once the semen is ejaculated here, and this it carries number of sperms. These sperms move towards the uterus and finally headed towards the mouth of this fallopian tube. Okay, after that the secondary oocytes after that, the secondary oocyte released from the matured Graafian follicle. Secondary oocyte released from the matured Graafian follicle of the ovary and enters into the fallopian tubes. Here they the secondary oocyte, it releases from here from the matured Graafian follicles of the ovary and enters inside the fallopian tubes, and where it has to fertilize within a period of 24 hours. Okay, though surrounded by several sperm, oocyte is fertilized by a single sperm. As we know, we have already discussed that the semen contains a thousand of sperms, but there is a one ovum to fertilize. So a number of sperms get surrounded. They surround the single ovum, and only one sperm is able to by capacitation to fertilize the ovum. Okay, now the so during meiosis 2 the sperm enters the secondary oocyte and completes the meiosis. After this, the secondary oocyte is known as the egg. As we know that the girl born with the primary oocyte in her ovary but arrested at a prophase 1 of meiosis 1, but at puberty there is a these primary oocytes again become functional and completes meiosis 2. Okay, so this very secondary oocyte releases from the matured Graafian follicle after uh, meiosis 1. Okay, so here this slide shows that the girl born with the primary oocyte in her ovary at birth. The girl born with primary oocytes, and later the primary oocytes arrested at prophase 1 of meiosis 1. Later at puberty, when the arrival of hormones is to be there, in the influence of these hormones, it completes its meiosis 1, and later got arrested at metaphase 2 of metaphase 2 of meiosis 2. Okay, so moving to the next. Now both sperm and egg can show their vitality only to a limited period. The sperm is alive for 48 to 72 hours in a female reproductive tract, and the egg can be fertilized for 24 hours before it is released. It is viable for a period of 24 hours before it is released from the ovary. Okay, now further moving to the next, and now coming to the steps of fertilization. What are the various steps that is to be involved during the process of fertilization? First is the acrosomal reaction, second is the cortical reaction, third is the sperm entry. So what is acrosomal reaction? The reaction during which the sperm reaches the egg and allows the sperm to break the surrounding jelly coat in is known as acrosomal reaction. As we know that the acrosome is present at the head region, at the portion of the head, and these acrosome contains different kinds of enzyme like proteolytic enzyme or acrosin. These enzymes helps for the these enzymes helps for the dissolve of to dissolve the jelly coat. Let's take a highlighter and we'll show you what is a where is jelly coat. So this is the egg cytoplasm. This is the egg cytoplasm, and egg cytoplasm covered with the membranes. Okay, so a membrane contains zona pellucida, then coordinated with this jelly coat. Jelly coat have move forward. It has a perivitellin space, and then within the cytoplasm there is a cortical granules. So this jelly coat is dissolved with the help of these proteolytic enzyme or acrosin enzymes present in this very portion of the head region of the sperms. As we know that the sperm consists of three parts, that are head, middle piece and tail. The head contains acrosome and this male nucleus. Okay, now this jelly coat once dissolved, the again the sperm move forward, and when it come in contact with this perivitellin space, so what is the next, this is the acrosomal reaction. Okay, when the sperm's head releases its proteolytic enzymes, acrosin enzymes, to dissolve the jelly coat for the move, so that it can move towards the cytoplasm of the egg. Next to this is next step is cortical granules, cortical reaction. So what happens in cortical reaction? As we seen here in this slide, let's take a highlighter to mark it and understand it clearly. Okay, so as we seen here that a sperm enters, it move forward, and once it dissolves the jelly coat, it come in contact with this perivitellin space. This is a perivitellin space. So this perivitellin space uh, really now when the sperm enters or touches the perivitellin space, then the cortical granules are present in the where they are present, cortical granules are present uh, there in the cytoplasm of a egg. And what happens after fertilization? An exocytic calcium dependent process occurs, which is referred to as a cortical response. The cortical response which prevent polyspermia, so that the other sperms, it prohibit the entry of the other sperm, so that only one sperm is able to fertilize the ovum. Okay, single ovum. So the mechanism brought on by release of cortical granules from the egg during the fertilization. This is characterized by the fusion of the cortical granules with the perivitellin space of the membrane. So here, as shown, that this is a perivitellin space, and these are the cortical granules shown in the yellow spherical dots. Okay, these are the cortical granules. They contain some material, and these cortical granules discharge, they fuses with this perivitellin space and form a kind of barrier as shown here. So this barrier ensures the prohibition of other sperms. Only one sperm enter inside the egg cytoplasm, fuses with the egg nucleus and prohibit the entry of the other sperm. So this is all about the steps of the fertilization involved in the fertilization of humans. Now here this slide shows the entry of a sperm nucleus. So as shown here that this very portion shows that how it come in contact with the perivitellin space, and then the male nucleus enters inside the egg cytoplasm, and there is a formation of what, there is a formation of this a barrier to prevent the entry of the other sperms. So in sperm only the nucleus is able to enter inside the cytoplasm of a egg. The middle piece and the tail portion remain outside of the egg or ovum. Okay, this is all about the today's video, and thank you.